I'm Heino Heyman, Director of Field Metabolomics Sciences at Metabolon. Welcome to episode 8 of our Molecules to Medicine series. Today we are tackling a critical safety concern in drug development called drug-drug interactions. We'll explore how metabolomics not only identifies these interactions but reveals the underlying mechanism and provides translatable insights that can guide safer therapeutic development. This next case study looks specifically at profiling of drug-drug interaction or drug-metabolite interaction, as well as the mechanism of action of a drug called probenecid. Probenecid is a FDA-approved drug that has historically been used for gout relief, but also to slow down clearance of drugs that were in short supply, specifically in the Second World War. Probenecid has also been used with several other drugs in this manner, exploiting what are now referred to as drug-drug interactions. Probenecid is also used as an inhibitor of several transporters of organic anions for in vitro studies in the research setting. And probenecid is also known for it being impacting 3 widely accepted in vivo renal targets, primarily expressed in the proximal tubules. That is OAT1, OAT3, as well as urate1. In particular for the OATs, the OATs are important hubs of multi-specific proteins that aid in returning the body to homeostasis following a perturbation. So with that, the study hypothesis became that given that the primary expression of known in vivo probenecid targets OAT1, OAT3, and urate1, the inhibition of these proteins would thus significantly alter the levels of key metabolites in signaling molecules, both in blood and urine. So let's dig a little bit deeper into the study. The study design was specifically looking at 20 healthy participants, of which were 14 women and 6 men, who provided both blood and urine samples at baseline and 5 hours after the administration of 1 gram of probenecid. Then they used untargeted metabolomics using Metabolon's Global Discovery Panel to profile both the plasma and urine samples. And the results revealed that probenecid treatment altered the levels of hundreds of metabolites for as well as having a very specific drug metabolite interaction with urate. So globally, looking at the plasma metabolomics data, they identified 1,234 unique metabolites. This also included probenecid. Of these, 354 metabolites were significantly elevated. Looking at the urine metabolomics, also run on our platform, just over 1,300 metabolites were identified, once again including probenecid. 622 of these metabolites were significantly decreased, with 113 metabolites significantly increased. So they then specifically wanted to focus on OAT1's substrates. And so when they had a specific look at these, they clearly saw a OAT-mediated transporter pathway being significantly enriched, which makes sense. But they then also specifically identified 97 metabolites that were significantly elevated in plasma, but also decreased in urine, indicating a very specific OAT-mediated clearance. These metabolites specifically included signaling molecules, antioxidants, as well as gut microbiome products, specifically some bile acids. In contrast to this, though, the urate1 mediated clearance was very specific, with urate being the only metabolite significantly decreased in the plasma and elevated in urine, consistent with an effect of renal uptake by urate 1. So they took it one step further and then developed some knockout mice from OAT1 and OAT3 knockout mice, and they followed that up with global metabolomics with metabolon as well. And so from that data, they identified quite a large number of metabolites changing again, but specifically 15 metabolites were significantly elevated in both knockout mice, with 6 being unique to OAT1, of which they were able to get 3 unique chemical structures, and 4 were unique to OAT3. So these are potential endogenous biomarkers of OAT function. And here are just some of those examples, being the OAT1 3 chemical structures, as well as OAT3 only the 4 that were elevated in the OAT3 knockout mice. So this group then also then went ahead and even went one step further and suggested a probenecid stress test, which is an assessment of the tubular function in kidney diseases, focused on the proximal tubular organic anion transporter capacity of kidneys of patients that are being treated. So measuring the changes of approximately 100 OAT substrates in plasma and urine before and after probenecid was provided can be used to provide a direct functional readout of tubular secretion potential, and something that is not captured by GFR alone. So really an additive test to show the health of the kidneys, especially before you go into a trial. So this clinically could be quite helpful in identifying tubular impairments earlier, especially in healthy individuals, seemingly healthy individuals going into a phase 1 or phase 2 clinical trial. All right, so to wrap up this this episode, I would want to just highlight that untargeted metabolomics was able to identify new candidate biomarkers for OAT function. As well, it helped to improve the monitoring tools that are currently available for kidney function and disease, as well as drug dosing tools for that. So the additional suggested probenecid test is quite a nice way of understanding tubular function in kidney disease, but also in healthy individuals that are going into a clinical trial. So easily to be conducted for these patients or volunteers that can then be tested to see what is the renal function. And finally, this is also quite a good example of the validation of an animal model for human translational studies. Okay, so thank you for spending your time and attention with us today and throughout the whole Molecules to Medicine series. As we close this series, we invite collaboration with Metabolon to integrate metabolomics into your programs, from translational biomarkers to patient stratification, to mechanism of action, efficacy measurements, and drug-drug interactions, just to name a few. Our team is ready to scope out the right study and deliver decision-ready insights to move projects forward. All episodes remain available on demand, so feel free to revisit any of the topics across the drug development pipeline at your convenience. We hope this series was insightful and directly relevant to active and upcoming programs. We hope we welcome feedback and what other topics would be most useful. Please share priorities and ideas for future sessions. We look forward to working with many of you and helping reach near-term research goals in the months ahead. Thank you for your time. We'll see you in the next series.